Okay, welcome to Ask the Expert. We have Xiaoyan. She is a PhD candidate in biomedical sciences at ULB, Center for Diabetes Research, University of Libre de Bruxelles in Brussels, Belgium. She has a background in molecular biology and bioinformatics with expertise in large-scale genomics data analysis. She started her PhD study in September 2019, supported by the Wallonie-Bruxelles International Scholarship for Sino-Belgium Outstanding Young Scholars under supervision from Drs. Miriam Knopp and Decio Azirik. Her research focuses on an integrative omics of human islets to gain insight into the pathogenesis of diabetes with a focus on glucolipotoxicity in type 2 diabetes and inflammatory responses in type 1 diabetes. And her talk today is mining the transcriptome of target tissues of autoimmune and degenerative pancreatic, beta cell, and brain diseases to discover therapies. So welcome, Xiaoyan. Very nice to meet you. And I can't wait to hear what you have to share with us today.
Yeah, I'll start. Thank you very much, Monica. Thank you also for the Super Science team for having me here. Today, I'd like to present the work that was recently published in iScience Journal, namely mining the transcriptome of target tissues of autoimmune and degenerative diseases to identify therapies. And firstly, I'd like to show you the rationale for disease selection. What common features do they have? For type 1 diabetes and multiple sclerosis are diseases of mistaken identity, where the immune system attacks and destroys our own target tissues. They share common genetic risk genes, chronic local inflammation, and target tissue damage. The target tissues are not bystanders of the autoimmune attack, but participate in a deleterious dialogue with the immune system that contributes to their demise. Several type 1 diabetes risk genes act at pancreatic beta cell level, regulating the responses to viral infections, the dialogue with the immune system, and apoptosis. For type 2 diabetes and Alzheimer's disease are degenerative diseases and show common amyloid deposition and chronic inflammation at their target tissues, pancreatic beta cell and brain, respectively. The striking gene expression similarity between pancreatic beta cells and neurons, such as expression of splicing regulators and splice variants. The same mechanism controls the expression of genes in both neurons and insulin-producing cells. So here we reach our research basis. Use one sentence to describe target tissues of autoimmune and degenerative diseases show signals of inflammation. We thus hypothesize that shared gene signatures can be identified from target tissues and mined for common therapies. Firstly, we collected RNA-seq data of pancreatic beta cells for type 1 and type 2 diabetes, the brain tissues for multiple sclerosis and Alzheimer's disease from public databases. Next, we integrated and compared the transcriptomes of pairwise diseases to identify the common signatures. Using these common signatures, we performed drug repurposing, and finally, we validated the drugs predicted using disease models for type 1 diabetes and type 2 diabetes. Let's look at some important features for the collected RNA-seq data. For type 1 diabetes, we got FACS-sorted pancreatic beta cells, while for type 2 diabetes, we are only able to access pancreatic islets. For the two brain diseases, we got uptake, chiasm, and prefrontal cortex for multiple sclerosis and Alzheimer's disease, respectively. And due to the different prevalence of diseases and the difficult access to target tissues, we got only 4 to 5 target tissues of individuals affected by type 1 diabetes and multiple sclerosis, as compared to a larger number of type 2 diabetes and Alzheimer's disease. All the cohorts have a well-balanced age range. And here, the last column displayed the number of detected genes in each study. there are more than 17,000 genes that were commonly detected in all studies, which gives us a good foundation for data integration. And since the rank-rank hypergeometric overlap analysis lies in the core of the present study, so I'd like to show you the principle of this analysis before stepping into the result part. Start from here, as we will compare pairwise diseases. So 2 independent profiling experiments are needed. We will perform differential gene expression analysis first for the disease individually to obtain the fold change for the genes measured in both experiments. And we rank the fold change from the most up to the most downregulated ones. And we next perform hypergeometric test to evaluate the significance of the overlapping gene or the intersected gene above each rank combination, since we will slide the rank threshold and irritate all the rank combinations. Via the hypergeometric test, we evaluate and determine the most significant overlapping rank index and point it out in this RRHO map. As you can see here, the aim is to extract the optimal overlapping gene set for downstream analysis. So for the present study, I use the first version of the RRHO analysis. Actually, our lab has developed an optimized new pipeline based on this version called Red Ribbon, which is more faster and more accurate. So if you feel interested with it, you can refer to this citation. So based on the RRHO analysis, we observed the global transcriptomic similarities, as you can see in the left panel. Let's look at the corner here. You can see type 2 diabetes and type 1 diabetes show the highest correlation in both up and downregulated gene programs. In addition, in type 2 diabetes and Alzheimer's disease, we also observed the similarity in both up and downregulated gene programs. While in other disease pairs, we only observed the similarity in the upregulated gene program. And the right panel displayed the number of commonly regulated genes, which corresponds to the left RRHO panel. For example, the 2,599 genes were commonly upregulated in the 2 types of diabetes, which corresponds to this RRHO quadrant. So due to the interest of time, I have selected several disease pairs as an example to be displayed here. In the first selected disease pair, you can see here the comparison between multiple sclerosis and type 1 diabetes, which are both autoimmune disease. In this plot, we identified 983 intersected genes, or we can call the commonly upregulated gene here in this corner. For the functional enrichment for those genes, the top enriched pathways are related to inflammation and adaptive immunity, such as the signaling by interleukins, interferon gamma signaling, interferon alpha beta signaling, and their downstream signaling, PD-1 signaling. Besides, we also enriched the antigen processing cross-presentation and the antigen presentation. In the second selected disease pair, let's say the Alzheimer and the type 1 diabetes. In this plot, in this comparison, we identified 658 commonly upregulated genes in this corner. And again, for the functional enrichment, to be here, you can see we got the very similar functional enrichment. Top enriched are related to the signaling by interleukin and interferon gamma signaling and PD-1 signaling. But here we didn't see the antigen presentation signaling. And in the last selected disease pairs, the comparison between the type 2 diabetes and Alzheimer's disease, which are both degenerative diseases. In this comparison, we observed 1,453 commonly downregulated genes here. For the functional enrichment, you can see the top enriched pathways are related to neuronal function, like the neuronal system, transmission across chemical synapse, neurotransmitter release cycle, and very interesting, the regulation of insulin secretion. Both pathways are quite functionally relevant. Based on the common gene signature via the RRHO analysis, we performed drug repurposing via the connectivity map analysis. So the CMap library was developed by Broad Institute, and which contains pre-computed gene signatures of 9 core human cell lines exposed to more than 25,000 perturbagens, corresponds to more than 19,000 compounds. In our cases, we adopted 3 critical steps. Step 1, to prepare the most regulated common signatures from RRHO in pairwise diseases. In the step 2, we compared our differential signature with the pre-computed signature in the CMap library. Step 3, to find the strong connection. In our case, we consider the most negatively correlated perturbagen as the potential therapies that could revert our gene expression signatures. And based on the CMap analysis, as you can see here, the heat maps, the top line above this heat map displayed the cells that were used to do the chemical compound or the drug treatment in the CMap library. And here displayed the tau score, which is the normalized connection score. The higher score means stronger connection. So you can see the CMap analysis unveiled bromodomain inhibitors as a top drug that could revert the gene signatures that were commonly upregulated in the most combination of the disease pairs. As you can see here, type 1 and type 2 diabetes comparison, and also the multiple sclerosis and type 1 diabetes and the other 2. Besides the drugs for the upregulated gene program, we also predicted the drugs that could increase the gene programs that were downregulated in 2 comparisons. One is from the comparison between the 2 types of diabetes, and another from the 2 degenerative diseases, let's say the type 2 diabetes and Alzheimer's disease. And very interesting, we identified in both conditions the JAK inhibitor. It is well known that one of the JAK inhibitor, paroxetinib, is presently being tested as the therapy for type 1 diabetes. So let's get back to the very promising predicted bromodomain inhibitor. Here we refer to one science paper which published 2 years ago entitled Selective Targeting of First bromodomain and second bromodomain of the BET proteins in cancer and immunoinflammation. What is the BET proteins? The bromodomain and extra-terminal domain family is composed of germ cell-specific and ubiquitously expressed members. They are components of chromatin remodeling complexes that promote chromatin decompaction and transcriptional activation. Let's call it the epigenetic readers. Please look at this schematic. So the BET protein can combine with the acetylated lysine amino acid residue from the histone to trigger the transcription of target genes, mostly are the pro-inflammatory genes. And if we block this combination with the BET inhibitors, there is no transcription for the target genes. Previously, the Gillen and colleagues validated that the second bromodomain inhibitors were particularly effective in models of inflammatory and autoimmune diseases, such as psoriasis and collagen-induced arthritis. And also, the Coley and colleagues also validated that The broad bromodomain, which targeting the first bromodomain and the second bromodomain inhibitors, IBET151 and JQ1, prevent interferon alpha-induced inflammatory genes in human islets and endothelial cells. So in our experiment, we aim to test two selected bromodomain inhibitor. Here, one is IBET151, which is a broad action target the first and the second bromodomain. Another is the bromodomain 2 specific bromodomain inhibitor called JSK046 in the models of human beta cell dysfunction in type 1 diabetes. So here, in our experiment, we exposed human islets or the human beta cell lines to the combination of two cytokines. One is interleukin-1 beta plus interferon gamma for 48 hours, which mimic the very advanced stage of type 1 diabetes dysfunction. So as you can see in the 4 plots, the major human compatibility complex class 1 genes and chemokine and cytokine and also ER stress marker CHOP were induced a lot by the treatment of the combination of cytokines, while the treatment of two bromodomain inhibitors could decrease the pro-inflammatory effects and ER stress induced by the treatment of cytokines. In addition to the gene level, we also assessed the protein level by the ELISA. We evaluated the secreted chemokine and the cytokine, and we observed the consistent result as observed in the gene level. That is, the protein level of CXCL10 and the protein level of interleukin-6 were decreased by the treatment of the bromodomain inhibitor, especially this one, the broad action one, IBET151. You can see here that in the bromodomain 2 specific one, we only observed the decrease for the interleukin-6. So we also validated the two selected bromodomain inhibitor in the model of human beta cell dysfunction in type 2 diabetes. Here, we exposed human islets to the 0.5 minimal palmitate for 48 hours, which mimic the metabolic stress in type 2 diabetes. As you can see here, we observed the gene expression of CXCL1 and interleukin-8 were decreased by the treatment of bromodomain inhibitor, especially the broad action one, you can see here and here. But there is no protection against ER stress was observed as we see in the type 1 diabetes model. In conclusion, we integrated the transcriptomes of target tissues from 4 major diseases affecting beta cells or the brain. The key inflammation-induced molecular mechanisms are shared between beta cells and the brain in autoimmune and degenerative diseases. We identified bromodomain inhibitors as agents that decrease islet inflammation in models of type 1 and type 2 diabetes. Last, drug repurposing points to other interest therapeutic candidates that could be further validated in preclinical disease models. Thank you very much for your attention, and I'll take your questions.
Yeah, that was great. Really, really exciting work. I think I wondered if anyone from the audience would like to have, a couple or ask a few questions. Okay, so then I have some questions. What prompted you to think of this, to sort of like begin this approach? What was sort of fueled this approach originally? Just the fact that these bromodomain inhibitors had success in other, inflammatory states?
First, as I mentioned in the background slides, because some like the diseases, like the autoimmune disease and the degenerative disease, as we selected in our present study, they have many common features from the molecular level. So I think it's deserved to identify the common therapies For
Right.
example, the type 1 diabetes also complicated with other autoimmune disease, and the type 2 diabetes also, the patients have a higher risk to get the Alzheimer's disease. So that's the start of our scientific approach.
Yeah, that's where you started to build. And then, when you're talking about, there was one JAK inhibitor, right? Baricitinib, this is just a hard tongue twister, that's being tested for type 1 diabetes right now.
Yeah.
How, who's doing that work? There it is, baricitinib. Is that associated with your group?
No.
Or is that on its way to the clinic, or what's the story with that?
So I think these clinical trials are under held by one company, but I forgot the name. But our group also do some relevant testing regarding the JAK inhibitor. Currently in our group, the Dr. Savatani is working on some drugs under this category, but still under testing.
Yeah, so she's trying to, or that lab's trying to kind of tease out which inhibitors may be, specific, right, for T1.
This one?
Yeah.
Yeah, we are going to test if use this inhibitor can recover the function level, like the insulin secretion. Also very interested about that one.
Okay.
Just a point to help Xiaoyan in one of the questions. This JAK inhibitor clinical trial is made by Ellen Thomas and Tom Kay in Australia, together with colleagues. We, for the time being, were more interested in TYK2 inhibitors in the context of type 1. This is what our data. And just coming back, your first question, Xiaoyan, was more, why did we select the bromodomain inhibitors? And I think this was an open question. So we just selected what the analysis gave us. So we went for the top. So it was a non-biased selection. Right.
That's what I was kind of getting at. I just wanted to kind of hear it said, that it was a non-biased selection. You basically, just to reiterate that you went panning for gold. You found the the targets, and then you went along with that. And then I would wonder, if you can comment, if anyone can comment on, what's the next step for, now that you've identified, sort of, the high level workings here. What's next step?
Next step, I think we can see other interested drugs here that we can try to validate, use in vitro model, and the bromodomain inhibitor, if possible, we can also do some other exploration, because the bromodomain inhibitor, bromodomain protein, the beta protein at one hand can combine to the promoter to make the decompaction of the condensed chromatin to shape the complex for the transcription initiation, and also they can include recruit the transcription factor. So if possible, we can do some exploration about the critical transcription factor, maybe involved in the mechanism. That's the direction.
I think Slavika, who has a nice work, has a question.
Yes, I have kind of both question of interest and little bit philosophical question on the premises of your strategy. And given that hypergeometric overlap test, when selected these overlapping up or down regulated genes between different morbidities, in some case here even comorbidities, like type 2D and Alzheimer's disease. But my question is, these subsets of genes that highly overlap, actually we cannot discriminate, given in the light of disease doesn't have abrupt onset, but it starts with adaptation and ultimately ends up with failed adaptation. How, what is the value of this approach, given that the hypergeometric rank based testing cannot discriminate between adaptive genes and maladaptive genes, or the cause and the consequence? So since you mine these genes and they are like a big pool of genes which are ultimately used for your drug discovery strategy, I'm interested, what is your thoughts on the value of this hypergeometric test to instruct you on your drug discovery strategy?
Thank you for your question. So maybe I can explain more about this part. So the hypergeometrics was performed here. So you can see the two independent profiling experiment and ranked by the fold change. And we will slide the rank and iterate all the rank combination, everyone accompanying the hypergeometric test, and to get the p-value, let's say the significance. And here we pointed out the most significant one. So as you can see here, so we define the genes intersected above this rank combination. So this one, if intersected, and here we go this strongest signal, we see those genes are commonly upregulated by the disease pair here.
Xiaoyan, I think Slavika knows, I mean, she has published a couple of nice papers using this technique. I think her question is a crucial one, because what she's saying is that we are finding common genes between these diseases, and we don't know if these genes are causative or if they are the response of the suffering cells to the inflammation, for instance. And this is a very difficult question to answer. What we will do together with Xiaoyan is try to obtain, and we have already obtained it, data sets at different stages of disease. So this study started several years ago. Now there is much more, including single-cell RNA-seq, and including studies where you can look at patients, let's say in the first 2 or 3 years of disease, and after 5 or 6 years. But the ideal would be to get material in the very early stages. And here we are limited by availability. But I think this is an important point. Anyway, even taking this into account, we already identified 2 compounds that may go to the clinic: TYK2 inhibitors, JAK inhibitors, and bromodomain is at an earlier stage. But your point is very well taken.
Thank you.
Yeah, I would extend this question or this discussion to ask, excuse me, if, say you get, to the clinic and to clinical trials, and you've got your bromodomain inhibitor or some other drug, how would that be administered so that it's not sort of a global effect? I mean, are you, is it going to be sort of a locally, just a thought experiment here. You've got someone in a prodrome, they've got the genetic risk factors and they've exhibited one autoantibody. Now, do you go ahead and, give this person or some kind of injection to the site, to the local area, or could you do this globally just for a limited time period? I mean, what does that really look like?
I didn't see any literature or anything about the clinical use for the bromodomain inhibitor, at least for the GSK046 and IBIT151. I only find some previous study mentioned they did some testing in use the mouse model, like the oral take one and also the injection one.
Mm-hmm.
Yeah.
Just adding a little bit, I think for the bromodomain, they are being used in cancer, and they are used systemically. I think that this drug is too non-specific, at least for the time being.
Yeah.
TYK2 inhibitors are used systemically in psoriasis, so they could be repurposed. Our dream is to target, and we have already discovered and patented at the lab 2 biomarkers that are beta cell or islet specific. And we are working with 2 companies to conjugate them to nanocarriers to target them to the beta cells. But I think this is work for the coming few years or many years.
Yeah. And just to your point about really dissecting into, that whole idea of adaptive versus maladaptive genes, when you're talking about the prodrome, you're looking at samples. What would be the best repository to get at, samples like that for you all? I mean, Enodia's got some things going. They've got the UK Biobank. We've got NPOD over here in the US. Where could you find those samples that you could really start to ask that question, like adaptive versus maladaptive genes? How can you, discriminate? That's a big question, but you need the samples, correct?
Yeah.
I think for Alzheimer, there is enormous amounts of samples, including at very different stages of disease, bulk and single cell. For type 1, what Xiaoyan has downloaded is a lot of data from the HPAP. So we are doing pseudo bulk analysis to increase the power. And there is enough samples to at least discriminate people with less than 3 years of disease and more than 7 years. So this is and type 2, there is also enough samples.
Well, that's good to hear that, that HPAP is collaborating with your group and to make that happen. It's very positive.
Well, they deposit their data, so it's all open access, and every week it's increasing the end. So they are really being a very, very useful source.
That's
I have
great.
one more question, if I may ask.
Yes, of course.
Yes. So another question is, I'm interested because I didn't pick up right. So in terms of suppressing or affecting the ER stress, how different is between JAK inhibitors and BET interacting? BET inhibitors. What is the difference and also in type 1 and type 2 diabetes? And I'm asking because I know that also in type 1 diabetes, ER stress is kind of prominent event.
Yes, I think in type 2 diabetes, because this induced by the metabolic stress, I think it's directly induced by the free fatty acid, the palmitate, something like that. It can change the ER shape and stimulate unfolded protein signaling. So, and then finally to stimulate the the apoptosis finally. And in type 1 diabetes, I think is more induced by the inflammation directly, I think. Yeah.
But what is the effect of your inhibitors or ER stress in context of type 1 and type 2?
So here,
what are your results? Yeah, in the models.
We actually tested the ER stress marker 12 and also XBP1, another one, in both type 1 and type 2 diabetes model, but we only observed the treatment of bromodomain inhibitor can decrease, you see here, decrease the ER stress marker in only type 1 diabetes model, but not in palmitate-induced beta cell dysfunction in type 2 diabetes. So I didn't show the plot here, but you can see here, we see this result.
What I think, just if I may say, maybe you don't have that aspect of ER stress in your model just with palmitate, that kind of mimics metabolic stress, but without component of proteotoxicity. So maybe you do not have actually the stress as such as in the size as will normally occur in presence of proteotoxicity. So maybe that's why you cannot measure big difference in type 2, in your model of type 2, when using only palmitate to induce metabolic stress.
Yeah, but I remember we a treatment of palmitate increased the stress marker. Yeah, but not only no protection from the bromodomain inhibitor.
I see. Okay.
Yeah.
Mm-hmm.
Well, I'd say we've had a great discussion here. It's a little bit past the 9:30 mark. So in the interest of everyone's time, thank you again, Xiaoyan. This is great. Really interesting work, and we cannot wait to see, what comes next from the laboratory. I think your group is really on to something quite interesting, and I love the collaborative approach you're taking. I would also say, best of luck, in the new year. And if anyone has any questions, I'd say reach out to this group, the Zurich group, and maybe to Xiaoyan herself. Thanks again, and have a great rest of the day.
Thank you. See you.
Bye-bye.